Okay guys, let's do this second half. We're going to start with A5, and then we'll do A4, which is like learning and innate behavior, and then A6, which is animal behaviors. Okay, but you already knew that because you've already been studying this ahead of time. Okay, so A5 is talking about neuropharmacology, so this is our drugs and like firing in the brain chapter. So first we want to start by talking about excitatory versus inhibitory neurotransmitters. Some neurotransmitters, when they're set in the synapse, they excite the postsynaptic neurons. So then that's kind of what we've usually talked about is you have a presynaptic neuron send a message to the synapse, hops across the synapse, the neurotransmitters hop across the synapse, knock on the door of the postsynaptic neuron, and the message continues. So most of the time we've been talking about excitatory neurotransmitters, but then there's also inhibitory neurotransmitters, which the presynaptic neuron sends neurotransmitters across the synapse, and then they go knock on the doors of the postsynaptic neuron, and instead of the message continuing, it shuts down the postsynaptic neuron. And that's kind of what we just talked about in the last video, which was with the rods and cones. The rods and cones, if they don't get any light, they depolarize and they send a inhibitory neurotransmitter, which shuts down the bipolar neurons. And if they do get light, they don't depolarize, so then there is no inhibitory neurotransmitter, and the bipolar neurons, they depolarize and then they send a message to the next guy. So like that's one example of inhibitory neurotransmitters. That's not bad. It's just the way that our body works. Okay, so I think I've just kind of covered that. So let's move on. Okay, so when you send an inhibitory neurotransmitter across the synapse, you form something called an IPSP, which is an inhibitory postsynaptic potential, which causes the guy that receives that signal to hyperpolarize, which is this like dip below the resting potential. So it gets more negative than negative 70, which means fewer signals are sent. Because remember, you have to become positive if you're going to send an action potential. And then what we've mostly talked about are EPSPs, which is excitatory postsynaptic potentials, which cause you to become positive, and then you send a signal. You propagate a signal down your axon. Propagate a signal down your axon, which then causes you to release more excitatory neurotransmitters. And then there's this concept called summation, where when you have an IPSP and an EPSP of equal magnitude, and they're being sent at the same time, they add together, and then maybe in this case we have an EPSP plus our IPSP, and it makes this little blip of a positive change of the millivolts inside of the axon, or maybe being received by the dendrites or the cell body. And it's a pretty small blip, so we don't actually meet threshold. And if we don't meet threshold, we don't have action potential. And if we don't have action potential, we're not sending a message. So then if we have many EPSPs kind of build up together, like if we had one here, another here, and another here, then they do all add up together, which we call summation, and then we get a bunch of action potentials. So we've discussed this before. Every stimulus that you get only makes the same action potential. So whether you get punched by somebody or whether you get tapped by somebody, the action potential only ever goes up to like positive 35 millivolts. And how do we differentiate between getting punched and getting tapped? So if you get tapped, you make fewer EPSPs, so you just don't have like this constant action potential firing. And then if you get punched, you make so many EPSPs that they all sum up together, and then your action potentials kind of go crazy, like we kind of see in this picture here. And then, of course, all the neurons are going to be hooked up to other neurons because of synapse formation. So sometimes you might be getting EPSPs from 3 of your synapse neighbors, and then an IPSP from another synapse neighbor. So these are all different examples of summation, and that's all you guys really need to worry about. Anything else I want to talk about? No, you guys should just kind of be able to be familiar with this idea. And then if you guys see EPSPs or IPSPs in like an image that will look like this, you guys should just be able to know what's going on. In addition to inhibitory versus excitatory neurotransmitters, let me actually add on to that. You guys will hear something called glutamate. Glutamate is an excitatory neurotransmitter, and GABA, GABA, is an inhibitory neurotransmitter. Just for an example for you guys to know, for life or Jeopardy or something like that. Okay. There's also slow and fast neurotransmitters. Slow-acting neurotransmitters, they hang out longer in the synapse, so like their footprint or their shadow is longer. So they're the ones that end up helping you with learning. And then fast-acting neurotransmitters might help you more with like an action or response, but the slow-acting ones, they hang out in the synapse longer, so they make that shadow, which aids in long-term learning. And we've already said that neurotransmitters are supposed to kind of act fast because we need to respond fast to stuff. So in the past, we've kind of just talked about we've just talked about excitatory, and we've just talked about fast, but now we're introducing the other side of things. There's also inhibitory, and there's also slow acting. Other things I want to talk about. Slow acting neurotransmitters are norepinephrine, dopamine, and serotonin. And so their effect is longer lasting in the brain. Fast-acting neurotransmitters would be like acetylcholine. That's the one that activates your muscles to contract. And the reason why slow-acting neurotransmitters are slightly different than fast-acting neurotransmitters is because they don't directly do the activation of postsynaptic neurons. They do this thing called second messengers, basically like a chain reaction, rather than being the direct guys that cause the change. But I don't think you guys need to know that in detail. So like we said before, slow-acting neurotransmitters, they are involved in memory and learning because they linger for longer and because they set off like a chain reaction in your cells. They can persist for days. We call that long-term potentiation. It's why we believe that studying is like repetitive. So if you study on Monday, Tuesday, and Wednesday, you'll get it better than if you just study one day, because you keep having those slow-acting neurotransmitters get released, and they still work days after they've been released. But that's all we really need to know. But I do want to mention hippocampus, because we didn't say that earlier. The hippocampus is like the memory, long-term memory part of your brain. And then I also want to talk quickly about endorphins. Endorphins are natural molecules that we find in our bodies that act as painkillers. So they will bind onto our receptors, and they will kind of be like inhibitory. So they'll stop passing the signal of pain. So then we don't feel pain. And then that one's natural. Endorphins are natural. But then synthetic endorphins would be like morphine. And then a lot of drugs will also kind of have like a painkiller inhibitory response. And then when we talk about like our opioid epidemic that we have in the US, also dealing with endorphins or molecules that are shaped like endorphins and bind to receptors of endorphins. And endorphins are one of those things that you make after exercise or when you are high on adrenaline. It helps to be like a protective factor from the pain that you have like after exercise. Okay, helps you to kind of overcome the temporarily painful stimulus. Okay, psychoactive drugs can increase or decrease postsynaptic transmission, so they can be excitatory or inhibitory. And the drugs that you guys want to know, you want to know at least 2 examples of excitatory drugs. Nicotine is excitatory. Cocaine is excitatory. Amphetamines are excitatory. So like meth is an excitatory drug. I think heroin is also excitatory. And then some drugs that people will take, like Ritalin, is excitatory as well for ADHD. Inhibitory drugs would be benzodiazepines, like Valium. So a lot of times they'll use that for people who are like schizophrenic, to kind of calm them down. But that's not the only job of benzodiazepines. Benzodiazepines can also be used for people who are like insomniacs to help them sleep because it's inhibiting like excess signaling. Alcohol is also inhibitory. Usually in really small amounts, alcohol is excitatory because of the glucose. So it's kind of like a sugar rush from breaking down the sugar in alcohol because it is a high carb beverage. And then with more than small amounts of alcohol, it becomes inhibitory. And then THC is inhibitory as well. Okay. Ecstasy, I would say, is excitatory, or also known as MDMA. It's going to impact serotonin and dopamine metabolism, so it basically kind of keeps those things in that synapse. So I think we might have talked about this in your class, but 17 and 18 are starting to blend together a little bit for me. So I would suggest you guys to take a look at this graph, and you guys will see a graph for sure on paper 3 that you have to interpret that has to do with option A. So just kind of be familiar with it. Anesthetics, which decrease pain, they would also be inhibitory. So then there's local anesthetics like lidocaine, and then general anesthetics that cause like full body unconsciousness, and they would use that during like surgery. Anesthetics decrease your awareness. Sometimes people will completely forget what happened, even if they were awake before or after the anesthetic was applied. And then sometimes people don't get enough anesthetic and can wake up during surgery. There's a case of that in Grey's Anatomy. I think that person had some sort of disease, or not disease, but some sort of like genetic variation where they broke down the anesthetic molecule faster. So they had faster metabolism, so then they used up the anesthetic and they woke up faster. Okay. We can skip drug testing because it's in the green box, but fun fact, I have been drug tested like 3 times since I've started nursing school. Never drug tested for teaching. Okay. Stimulant drugs. Stimulant drugs are going to be excitatory. They mimic the sympathetic nervous system, which is our fight or flight system. They make you more alert, energetic. They can make you more self-confident. They're going to increase your heart rate, your blood pressure, your body temperature, probably also your respiratory rate a little bit. And then we'll have mild stimulants like caffeine in coffee, and theobromine in chocolate. I don't think you guys really need to worry about that. And then we have sedatives, which are the opposites of stimulants, which will be inhibitory. So like we said, GABA is inhibitory neurotransmitter. So it does come up in the book. I don't see glutamine here, which we just talked about, which is an excitatory neurotransmitter. And then they're talking about pramipexole, which I think is a drug. It's used for Parkinson's disease, and it probably increases how much dopamine is in the postsynaptic membranes. So it mimics dopamine, binds to dopamine receptors, because when you have Parkinson's, you don't make enough dopamine, which causes some of the muscle issues that we see when people have Parkinson's, like the tremors. And cocaine uses the same synapses that dopamine does. It binds to the dopamine reuptake transporters, which we've mentioned before, and because it's blocking those transporters, dopamine builds up in the synaptic cleft, and it keeps knocking on the doors of the postsynaptic neuron. Because we said most of the time what's supposed to happen is neurotransmitters hop across, they knock, they detach, they either get broken down. For example, acetylcholine gets broken down by acetylcholine esterase, or they get reuptake, like dopamine gets sucked back into the presynaptic cell. In this case, cocaine stops them from getting sucked back into the cell that they came from, so they're constantly knocking on the doors of the postsynaptic cell. And then Valium binds to an allosteric, which is like a different site on the GABA receptors, and causes inhibition, which is what causes people who take Valium to like calm down. So it's like a sedative. THC binds to cannabinoid receptors in the presynaptic membranes. This causes you to release excitatory neurotransmitters. It causes you to inhibit the release of excitatory neurotransmitters, which makes it inhibitory. I knew that didn't make sense. Okay, so THC binds to receptors, which stops the release of excitatory neurotransmitters, which makes it inhibitory. Sorry, not sorry for not knowing that much about drugs. Okay, so moving on. Okay, next, drug addiction we know is we know that it has some sort of genetic component to it. Also has environmental components, and many drugs are going to deal with dopamine synapses, because dopamine is the neurotransmitter that is associated with pleasure and reward. So it makes sense that addictive drugs would kind of prey on that neurotransmitter and cause you to have more of it. So it's going to cause you to keep doing the thing that causes more dopamine between your synapses. So like we said, genetic component, gene DRD2, which has to do with the dopamine receptor protein. Addiction is also dealing with environment, peer pressure, poverty, traumatic life experiences, mental health problems, all of that, cultural traditions, and then addictive drugs, opiates, cocaine, nicotine, alcohol. They all impact those dopamine secreting synapses to basically reward you for consuming those drugs. Okay. What I want to do now is go backward, and we're going to take a look at innate and learned behavior. Okay. Innate behavior we know is going to be inherited from your parents, and you're going to be born with these behaviors. You don't have to learn them. Sorry, I'm cleaning off my touchpad. And behavior can either be innate or it can be learned. So innate would be like reflexes, or when they're talking about how if you put like your pinky around a baby's hand, a baby is going to grasp its fingers around your pinky. And we call that the palmar grasp reflex. And we also have reflexes like on the bottom of a baby's foot. It's called the Babinski reflex. So if you like drag like a pen or something up along their foot, they will stretch their toes backwards, like towards their body rather than away from their body. Anyway, innate behavior is genetically programmed and it's inherited, so you're born with it. And skip the purple box. But you can do behavior experiments involving taxis. So you guys know phototropism and gravitropism, movement of plants towards light and towards or away from gravity. Taxis is animal movement towards stimulus, so like towards light or towards like moisture or towards heat or something like that. Kinesis is just like random movement. And taxis is more directed movement. But they might ask you guys to set up an experiment having to do with these two. I'm not sure they would, but you just want to know what those terms are. And that you need one independent variable, and you can have different levels of that independent variable, but it's easier if you just do one independent variable as your experimental group and none of that independent variable if you're going to do the control. And then you need to have a quantitative, if possible, and then qualitative, if not possible. So numerical versus descriptive dependent variable. do like 50 trials, because this is a pretend experiment that you would be setting up anyway, so go ahead and do 50 trials. And then you need like at least 3 controls. Reflexes are autonomic, involuntary, and they are innate. And we've already talked about that, so I'm going to move on. Reflex arcs we've talked about as well, involving 3 neurons and a receptor and an effector, like the withdrawal reflex, which I feel like you guys should know super well because we're constantly talking about it as an example. Reflex arcs show up a lot on paper 3 for option A. So like we said, you always have receptor cells like a photoreceptor or a thermoreceptor. They're always linked up to a sensory neuron. A sensory neuron is then going to link up to the interneuron or the relay neuron. And if we're talking about reflexes, it has to be located just in your spinal cord. And then the interneuron or the relay neuron is going to be linked over to a motor neuron. Motor neuron exits the spinal cord and it's attached to an effector. In this case, an effector is a muscle. And we would call this like a motor end plate where acetylcholine would come out and it would cause your muscle to do something. The other thing is we could see that the cell body of the sensory neuron is inside of the spinal cord. It's not hanging out here. And the cell body of the motor neuron is inside of the central nervous system, which is this bluish part. The relay neuron lives completely in the central nervous system. The sensory neuron lives kind of out of peripheral, and well, this is peripheral. I would say this yellow part is peripheral, and this blue part is central. And the motor neuron is inside of the central nervous system and comes out into the peripheral. Anyway, in the past they've had you guys label this as an image, so just know this, but you should, because we talk about this reflex arc all the time. Learned behavior is going to be behavior that results from experience. You are not born with it, and it will involve slow-acting neurotransmitters, like we talked about, like dopamine and serotonin. And then birdsong has to do with learning. It also has to do with being innate. So you are born knowing a birdsong, but then you can also pick it up from your parents. So if you have like a mockingbird and it's raised by a blue jay, it might learn a different bird song, but it was also born knowing how to do a bird song as well. And they've done some research on this, so we have a database question which shows a bunch of different notes of bird songs and kind of shows, I think, from what I recall, I don't think you guys did this one, but 17 did, just like merges of bird song, and it kind of feels like they're like ad-libbing some parts of the song. Anyway, but that's what this is. You might want to take a look at this database question. It's a pretty weird one. And you guys will have at least one database question on paper 3 to answer. We also have reflex conditioning, which is kind of like learning. It's a little bit in between like innate behaviors and learning. It's when we're able to associate a stimulus with a behavior. So this would be like Pavlov's dogs. So when they see the food, they salivate, that's a reflex, but then they can link a ringing of the bell with food coming, and then the ringing of the bell causes salivation, even if food is not available. In the case of the textbook, what they're talking about is birds having an innate reflex to avoid foods that have a bitter taste. So that's just something that they're born with. They eat something. They don't have to eat something to know that it's going to have a bitter taste. They're kind of born with it. But then they are going to be conditioned to learn which things have that taste. So if you eat something that is black and yellow, and that's a song, I think. Anyway, if they eat something that is black and yellow, and it doesn't taste well, it doesn't taste good, they're going to stop eating things that are also black and yellow through association. And then we already talked about Pavlov down here. You guys should generally know Pavlov, but it's a pretty well-known experiment. Plus, again, as we're constantly mentioning AP and IB Psych. Imprinting is something that has a particular window of time that you can learn it, and it's usually pretty early after birth. This can also be when we call it a critical period. So there's like a critical period or a critical window, for example, of like language learning, which we say usually is probably best to do like before 5th grade. So that's why we have those like elementary school immersion programs. But in particular, in the wild, we have imprinting for geese when they are born. It's like the first object that they see, they will follow around, and so they are imprinted with the idea of who their mom is. And this was done by Konrad Lorenz. So he got birds to follow him around. And this happens, he found out within 16 hours after hatching. And then if they meet their mom like at the 17th hour, they're not imprinted, so they don't just follow her around. So imprinting is learning because you're not born with it, but it's learning that requires a specific window. And then reflex conditioning is learning because you're making associations, but it's a very simple type of learning. Operant conditioning is trial and error. So there's an example where there's like a mouse or a rat that is in a little box, and there's 2 buttons. And one button causes the mouse to open like this trapdoor where food comes out, and the other one causes like a shock. So then once you push the one with the shock, you learn, I'm not going to push that one anymore. You push the other one and it gives you food, you're going to push that one all the time. So that kind of idea. We do a lot of operant conditioning with simple animals. So maybe like teaching them how to do like a maze or a puzzle or something like that, so they can learn trial and error. Again, a form of learning, but still a pretty simple form of learning. And anytime we talk about learning, it's just a new skill or new knowledge that they can use. And that can be associative learning, like the reflex conditioning, or it can be imprint learning, or it can be operant conditioning, trial and error learning. Okay. I'm going to skip the rest of this paragraph because I don't think you guys really need to worry about it. And learning is also going to involve memory, because if you cannot remember what you have learned, then you haven't learned anything. And we convert things from short-term memory, which is in our frontal lobe, and it moves to long-term memory, which moves to the back of our brain, where our hippocampus is. And the importance of the hippocampus was strikingly demonstrated in 1953 when a patient called Henry Molaison had his amygdala and a section of his hippocampus from both of his cerebral hemispheres removed in an experimental attempt to cure epilepsy, and he immediately became incapable of making new memories unless they were procedural, and his recall of memories formed during the 11 years before the surgery was also impaired. Experiences cause large number of new synapses to be formed, so when you learn something, you make new synapses, and that helps you to remember it. And then when you start to stop using that information, it might be pruned. Okay. And let's move forward and let's do the last one. So close to being done. Okay, last one is ethology. Here you guys just want to know the different examples of all the animal behaviors. Okay, ethology, study of animal behaviors, and then we usually want to talk about how it's advantageous to them. So natural selection has led to specific animal behaviors that we see today, because animal behaviors should be adaptive. They should be good adaptations and not just variations. Otherwise, they're probably not going to persist for a very long time. But then animal behaviors can also change based on the environment. So the example they're talking about here is a house finch, and the native population in California is sedentary, so they just hang out in the same area. And then a bunch of them were taken over to New York City and released, and then they became migratory birds within 20 years. So that's an animal behavior that they learned to do because it gets cold in the winter, and they wouldn't have survived. So the ones that learned how to migrate lived, and the ones that didn't died. So there's our natural selection there, and how that animal behavior can change. They talk about another bird called the great tit or the Paris major. Hopefully you didn't hear my stomach just rumble. The bird lives in the woods. It feeds on caterpillars and other young insects. The food capacity rises in the spring after we have a bunch of new leaves and spring has blossomed and all of that. But because of global warming, the time of peak availability has become earlier because spring and summer kind of come earlier and earlier. So they have had to kind of change the timing of their nesting and their egg laying. So they keep laying their eggs earlier and earlier to meet up with the time that the insects and the trees are blossoming. Okay, breeding strategies in salmon. You guys want to know the hooknoses and the jacks. They're types of a coho salmon species. And when you're a salmon, you do external fertilization. So the women spray their eggs into the water and the men spray their sperm into the water. And then they should meet up and they should fertilize and become zygotes. If you are I want to say hooknose. If you're hooknose, you fight with another male, and then whoever wins gets to spray their sperm onto the female's eggs. If you are a jack, you are small, and you don't really want to fight a hooknose. So basically what you do is while the males are fighting, you sneak up on the woman who is spraying her eggs out into the ocean, and you spray your eggs on them first. So the jacks are sneaky because they tend to be smaller. The hooknoses go directly and fight each other for the honor of being able to spray eggs or spray sperm on the eggs. And they found that occurs when males that grow rapidly tend to be jacks, and then males that grow less rapidly tend to be hooknoses. They grow slower, but then they end up being larger. And then jacks have like an early growth spurt, but then they're smaller, so it's just advantageous for them to have that kind of behavior. We also know about synchronized oestrus, which is when all the female lions in a pride, they basically have their menstrual cycles together, and it increases their survival and the reproduction of their offspring. So the way that lions work, when male lions are older, they are all expelled from the pride, so about like 3 years old. And then they go and they can either stay in their pride or they can go find another one. But in order to stay in their pride, they have to fight another male who wants to also be in that pride. And whoever wins gets to mate with all of the females in that pride. So when a new male comes into a pride, he might kill all of the cubs. And that makes all the females basically have their menstrual cycle or oestrus, and the male can then mate with all of the females because they're all menstruating or ovulating, and then all of the cubs that are going to be born in that next cycle are going to be his, and then they all get to live because they're his, and they all also get to leave the pride at the same time when they're 3, so they don't have to compete with each other, or compete with, I guess, like new guys coming in, because they're the ones that are all going to leave at the same time. I think that's all we want to talk about. Oh, also, the nice thing is that since all the females are giving birth at the same time, they also all lactate at the same time, so then they can all feed each other's children when maybe like another female is off hunting or something like that. So it makes it convenient for them to take care of all of their kids at the same time. Okay, next we have the black-cap, Next we have black-cap migration. These are the guys that have learned to travel. So black-caps that lived in Germany always migrated to Spain and Portugal for the winter because it's warmer and there's more food. And then we started to find that there were more black-caps moving over to Britain and Ireland instead of going to Spain and Portugal. And global warming has made Britain and Ireland warmer than Spain. Well, not warmer than Spain and Portugal, but warmer than they were before. So it's helped them to survive there. Okay. You don't have to worry about this experiment. You might want to look at it if you care. Vampire bats do this thing called altruistic behavior, which is anytime you do altruism, it is when you do something that might put you at a disadvantage, but it helps your overall group. So in this case, vampire bats drink blood, and if you don't drink blood for 2 or 3 days, you die, or you could die. But this doesn't often happen because bats that are able to go and find blood, they can do this regurgitation, which can feed the bats that could not eat enough of the blood. So it's called altruism because if you have eaten enough, you are giving up some of your food stores to your other bat friends that haven't eaten enough so they can sustain their bodies for a little bit longer. So it puts you at a slight disadvantage, but it puts your whole entire group at a better advantage. So we call it altruism. And we see that a lot with humans. We're a pretty altruistic species, despite sometimes not feeling like we are. But other species are much less altruistic. Then we also have foraging behaviors in crabs. So basically, they have some sort of system that they have learned where they can determine the amount of work that's going to be done and the amount of payoff that you're going to get. So shore crabs can eat mussels of intermediate size when they are in an aquarium. And there's equal numbers of every single size. They're going to pick the ones that are in the middle of that bell curve. And so they have a tendency to figure out what's going to give us the best payoff. Because the big ones, it might take too long to open up their shells. So too much effort for payoff, but a lot of effort. And then the little ones, they're easy to open, but they don't give enough payoff, so they end up eating the ones in the middle. So it's kind of like, I don't know, studying a little bit to get a B, or studying a lot to get an A. I've kind of been doing that this semester, just kind of being able to gauge how much effort you want to put in for how much payoff. Okay. And then birds of paradise, they do courtship based on how they look. So it also works with like tail feathers of a peacock, or like dances and courtship displays. So birds of paradise live in New Guinea, and they have very showy, well, the males have very showy plumage, which is going to be like their feathers. And the females build the nest, and they are not going to be as showy looking. So then the birds of paradise will do like a courtship dance to attract the females, because the females have the house, and they're going to also be the ones that you want to mate with. So you're basically trying to do a dance, so the lady will let you into the house. So apparently the males, they will gather at a site called a lek, and the females will select a mate from the males that are there. And it'll be based on how they look and their dances. And then Darwin's explaining that it helps with mate selection. So females prefer mates that have exaggerated traits, so then every generation the birds of paradise get more and more exaggerated looking. And then if a bird has to use enough energy to grow a plumage that looks elaborate enough and have these very vigorous courtship displays, then he must have been fed efficiently. So we're basically saying it's energetically expensive to have such nice feathers and to be able to have the energy to do a dance. So it kind of shows the females that if you have such a nice set of feathers and you can dance so well, that must mean that you have been well fed, and that means you probably have good sperm. So yes, I will mate with you. You can come to my house. At the same time, it also means that you might be good at running away, because if you're so bright and showy and fancy, predators probably want to eat you, but they didn't eat you, because if they had, you wouldn't be here doing this dance. So it's kind of indirectly pointing to females that that guy is a pretty good catch or something. So natural selection causes those traits to become exaggerated. And then we know that learned behavior can be spread and it can also be lost, and innate behaviors are more stable over time because they're genetically based. Learned behaviors then have to be passed down directly from parents to offspring through teaching. And if they're not, that's why they can be easily lost. So chimpanzees using tools, they also do this learning where they can, like, they do modeling. So if a chimp sees another chimp do something, it can kind of repeat that action pretty well. But if it doesn't see another chimp doing something, then maybe that learned behavior is going to be lost and not passed down from generation to generation. So the example they want you guys to know is blue tits and cream. So again, we're talking about birds. In Europe, in England, they used to deliver milk bottles in these little glass things with a cap on them, and blue tits would learn how to peck through those caps and then drink the milk to be able to use that as sustenance. And then there was a period of time when the Germans were occupying that area, so then they stopped delivering milk, and the blue tits learned to stop drinking or going to the houses to try to drink milk because it wasn't there. But then once the deliveries of milk started again, they popped up again. Yeah, they popped up again. But we don't see this behavior as much anymore because now people prefer like skim milk, it doesn't have as much cream at the top, and also we deliver things in supermarkets and not to outside of people's houses. So learned behaviors can be passed down, they can resume, and then they can also disappear, and then be relearned or not relearned, and so on. So you guys want to know for this chapter the different examples. They're not going to ask you guys examples that are not in this textbook, but it's also okay if you watch a lot of Animal Planet, if you know an example pretty well. Technically, it's okay for you to talk about those if you can't remember one from the book, but I would definitely advise you to try to go off of what you know from the textbook. I think that is all I want to talk about for now. So I will see you later.